and whenever you're ready.
So I don't usually say this, but I'm really excited about right now. So I get to talk to you guys about one of my favorite topics in immunology, and that's vaccine design. And I picked this topic for a couple of different reasons. I'm really interested in world history, weird stories, and seeing science impact people's lives. And I think in this presentation, you'll see the vaccine research has a lot of all three of those elements. So just to give you the general roadmap, in this presentation, we're going to try to build up the story of the past, present, and future of vaccine research. And so we're going to use this timeline to kind of orient ourselves and see how the field has changed over literally centuries, if not millennia. So I want to start way back, 3,000 years back, with kind of a cold case. So you're looking at the remarkably well-preserved body of this young man. And there's not a lot that we know, but there's some things you can see right off the bat. The high cheekbones, the sharp, angular features. If this guy was walking down the street, you'd probably notice him, is what I'm trying to say. So everybody meet Ramses V, pharaoh of Egypt, 3,000 years ago during the 20th dynasty. He ruled for about 4 years before his untimely death, and we don't know why he died. There are no hieroglyphics on the subject, but there are clues. And if you look along his right cheek, you can see these raised bumps, these sort of scars or lesions. And it's not clear exactly what that is. Maybe he didn't know how to shave, or maybe something happened. So the working hypothesis is that Ramses is the first recorded victim of smallpox, a disease that completely changed the landscape of the ancient world, killing 30% of people who were infected. And as you can see, it didn't matter if you were rich or poor, a peasant or privileged. Smallpox is part of the reason why the Roman Empire declined towards the 1st century AD. It's also the reason why Hernan Cortez and 300 soldiers were able to conquer the Aztec Empire. That doesn't happen if not for epidemics that spread throughout the New World. So this was a serious disease that our ancestors had to deal with. And by the 10th century, it seems like someone's onto something. So this is kind of a very primitive protocol from ancient China in the 10th century, describing a process called variolation, where you can take live infectious material in a controlled manner and protect someone against the actual disease. So there were different ways to do it. You could take scabs from someone who had smallpox, turn that into a fine powder, blow up a person's nose, some kind of mucosal challenge, if you want to think about it that way. You could wear clothes that somebody who had contracted smallpox had worn. There were different techniques, but at the end of the day, they found that you could improve outcomes and protection against full-blown smallpox. So variolation caught on. It started in China, but by the beginning of the 18th century, it had spread to India, the Ottoman Empire, and we also know to parts of Western Africa. But by the turn of the 18th century, variolation hadn't quite made it to the West. It hadn't made it to Europe yet. And so for that, we can thank Lady Mary Montagu. So Mary Montagu lived a pretty charmed life. She was a member of the British aristocracy. She was wealthy. She was known for her writing, for her humor, and for being beautiful, until her face was charred by smallpox, which also claimed the life of her brother. So she wasn't happy about that. But she had the opportunity to be exposed to a technique that really blew her mind. Her husband was an ambassador, a British ambassador to the Ottoman Empire, so she spent some years in Istanbul. And while she was there, she saw her first glimpse of variolation. So this is a journal entry that she wrote back to a friend in Britain. And she writes, "The smallpox, so fatal and so general amongst us in Europe, is here entirely harmless by the invention of engrafting," which is the name they give it. So engrafting, or variolation, was such an exciting thing to her that she rushed and got her 6-year-old son variolated. She then tried to have her physician bring the practice back to England. There was only one problem. A lot of people in England didn't trust variolation. They'd only heard about this being done in faraway foreign countries, and at this point on a couple of kids. So they wanted to see some firm evidence that variolation was safe and effective. But how do you test that? How do you variolate people and get them to sign up for that study, knowing that there could be danger? I mean, you'd almost need to have people that were willing to die. Yeah, that's exactly what happened. So the British government recruited a cohort of prisoners who were on death row for various crimes. These were young men and women who had stolen horses, cows, large sums of money, even powdered wigs. So basically, the king gave them the option to either serve out the punishment for their crime or sign up for the study. They signed up. And the idea was they were very elated. 5 of them developed mild fevers, flu-like symptoms, and they were then monitored for the following month. Everybody turned out to be okay. Some of these people were subsequently challenged with live smallpox and were still protected. So this actually goes down as the first controlled clinical study, a clinical trial in Britain. So there's still the question of that one guy who didn't have even a mild reaction to variolation. What happened there? So it turns out that the British government was looking for prisoners who had never had smallpox before. Otherwise, you're going to have pre-existing immunity. We can't test if variolation works. So this guy had lied his way into the study. Understandably, he didn't want to hang. And ultimately, he was allowed to go his way. So overall, it was a successful demonstration that variolation could work. But at this point in history, even after this Newgate prison experiment, we're relying on a technique that still has dangers. I mean, you're giving somebody active infectious material. 2 to 3% of people who were variolated died. On the flip side, 30% of people who got smallpox died. So it was still better in that sense. But doctors didn't really understand how to variolate. They were saying that you had to be bled by leeches extensively. They would say things like, the deeper the inoculation is into the tissue, the more effective it will be. Ultimately, this caused secondary infections, bacterial infections. And as a result, variolation wasn't really an entirely safe technique. So I want to interrupt this presentation for a brief public service announcement about milkmaids. Namely, they're absolutely beautiful. Am I the only person seeing this? All right, fine, don't believe me. Why don't we look at the literature and songs of the English countryside during the 17th century? So here's a song by a ballad by Martin Parker. Their labor doth health preserve, no doctor's rules they observe, while others too nice in taking their advice look always as though they would starve. So this is one of many songs and literary items praising the strength and health and beauty of milkmaids. And this was a well-appreciated fact in the English countryside. No one knew exactly why, but the working idea was that maybe it had something to do with the close contact to cows. So Edward Jenner, who was trained as a surgeon, as a clinician in London, but ultimately went back to the southwest English countryside to set up his practice, would have been exposed to ideas like this. And so he would have heard the rumors that cowpox, which is closely related to smallpox but induces a much less severe disease phenotype, could confer protection. So Edward Jenner, as I'm sure you all know, is famous for his really groundbreaking work in describing the nesting patterns of the cuckoo bird. So some of you know cuckoo birds are parasitic birds. They don't actually raise their hatchlings. They lay their eggs in the nests of other species. And as soon as those hatchlings hatch, the first thing they do is try to push over the eggs of the species they're parasiticizing in order to soak up the nutrients and affection and maternal care of the mother bird, of the unsuspecting mother bird. But really, you're probably more familiar with Jenner's vaccine work. So Jenner noticed that when Sarah Nelms, a maid out in the countryside of Berkeley, developed cowpox, there was an opportunity to actually test the hypothesis that you could protect people against smallpox. So he took his 8-year-old gardener by the name of James Phipps, inoculated the boy in the hand, in the upper arm, with cowpox, and then challenged James a couple times with live smallpox. James was fine, thankfully. Jenner then repeated that experiment on a couple different individuals and published the results and reported them back to the British Royal Academy of Sciences. We don't know the name of the cow in this study, but definitely an unsung hero of early vaccine research. So Jenner's work was met with mixed reviews. Namely, wait, you're putting cow material into people? So there are all these political cartoons suggesting, all right, so the guy who came up with this theory for cuckoo birds is now telling us to put cow material in our arms. Wonderful. Now we're going to turn into cows. The other criticism came from the medical establishment, because a lot of doctors had practices that depended on variolating people. And so this new rival technique was a threat to their ability to practice variolation and make profit that way. But ultimately, Jenner spent the rest of his life promoting and defending vaccination. which eventually supplanted variolation as a means of protection. And of course, vaccine comes from the Latin word for cow, vacus. But at this point, our vaccine still requires access to cows, to cowpox, which, if you're a doctor inside of the city, is difficult because you don't have easy access to materials in the countryside. So it's a less controlled and less efficient way of vaccinating people than what we do today. So really, we can thank Louis Pasteur for bringing vaccines out of the stable and into the laboratory. So Pasteur was culturing chicken or fowl cholera, and the story goes something like this. So Pasteur goes on summer vacation, and he entrusts his lab to a couple of assistants, and the last thing he tells them is, look, before I come back, just make sure you take the cholera cultures and inoculate the chickens, all right? They forget. The cultures get cold. The cultures get old. Pasteur comes back, and they suddenly remember to do it again. The problem is, the chickens don't die. At first, they think it's a fluke. So they go back, they get some fresh cultures, they re-inoculate the chickens, they're still protected. So Pasteur, who you know for the quote that chance favors the prepared mind, recognized that there was something interesting going on here, that maybe attenuating, aging, weakening microbes, and then immunizing people with those could protect them against full-blown infection. And ultimately, he used this strategy against other diseases as well, including rabies and anthrax. Rabies is particularly important because the vaccine is still really the only effective way we have to protect you. If you get infected, start presenting symptoms, literally the only other strategy for saving your life is to put you into a medically induced coma and see if your immune system can fight off the virus. And 6 out of 7 times you still die. So the vaccine is a pretty important tool here. In addition to developing vaccines, though, Pasteur had some other pastimes, like disproving this millennia-old idea you could spontaneously generate life from inanimate objects. Maybe some of you have heard this before, but there are all these recipes that I guess you can try at home for taking damp clothes and wheat, leaving those in the shed, and you'll get mice. Or the Romans thought you could take meat and flowers, leave them in a shed over the summer, and you'd get honeybees. So Pasteur showed that, no, you actually need living things to make other living things. So that was a really basic idea, but also a fundamental one for science. But what Pasteur didn't understand was how immunizing protected people. What are we doing here that's actually conferring protection? He had an idea, though, and his working hypothesis was that when you get infected with a microbe, that microbe consumes certain essential nutrients in your body that it needs to survive. And if you survive that infection, those nutrients are gone. So if you get infected again, there's nothing for that microbe to eat. I actually kind of like this hypothesis. It's simple, it's elegant, it's completely wrong. But without an understanding of how the immune system works, I think this is really the best idea for immunity that he could have had at the time. So here we are closing out the 19th century. We can make vaccines in labs, but we don't really know how they work. And so for that, Adolf von Behring deserves some credit. So von Behring was a German researcher who was inoculating guinea pigs with low doses of diphtheria toxin. And what he noticed was that if you did this, and you collected the serum from these guinea pigs, and you transferred it into a different guinea pig, like one that wears a Santa Claus hat, and you tried to challenge that animal with a high lethal dose of the toxin, that the guinea pigs actually survived. So that was success, or Erfolg in German. But what he didn't know was what was really in the serum. There's got to be something against this toxin, some antitoxin. So he teamed up with Paul Ehrlich, another German researcher who was developing this receptor or side chain theory of immunity. And I know Erica is also going to talk about this more. But essentially, Ehrlich put forth that cells had these side chains that eventually could also become soluble and bound in very specific ways to different molecules, toxins and maybe other foreign molecules associated with pathogens. We call these things antibodies now. And the studies that Behring and Ehrlich did together were some of the first experiments showing that passive immunization, transfer of antibodies from one individual to another, could be protected. So people started working and thinking about how you could more efficiently generate these antibody responses. So Gaston Roman, who was a French veterinarian at the time, noticed that you could inoculate horses with diphtheria toxin, inactivated toxin, and generate antibodies. But really, the horses that made the most antibodies, that had the most robust responses, were the ones that had a little bit of inflammation at the site of inoculation. So he had this idea that maybe you could help or adjuvant a vaccine by inducing sterile inflammation. So he started throwing the kitchen sink at these horses. Breadcrumbs, tapioca, various ions. This raises a couple basic points. Number one, what? I mean, what was this guy doing, right? The adjuvant design was not a rational process. But ultimately, these types of experiments led to the first popular adjuvant that we still use today, which is a preparation of alum hydroxide and other types of alum adjuvants. So, 1920s, beginning to think about how to make vaccines better, but maybe not in the most systematic way possible. And so that's really where we are going into the mid-20th century, the mid-1900s. And what really helped to foster a golden age of vaccines is something that most of us do every day and completely take for granted, which is the ability to grow cells in culture, and thereby the passage and propagate virus in vitro. And when you can do that, you can make virus adapt to different conditions. You can make it adapt to colder temperatures, warmer temperatures, different chemical conditions. You can attenuate the virus, as opposed to doing what Pasteur and Jenner had to do, which is go to organisms like cows to get cowpox. The vaccine against rabies depended on taking the spinal cords of infected rabbits and drying them out. So not really high throughput efficient ways to vaccinate people. So ultimately, being able to grow cells in culture, as basic as that sounds, spurred a golden age during which we were able to develop vaccines against measles, mumps, rubella, polio, all within fairly defined span of time. So that was the 50s and 60s, working our way up the timeline. I now want to give a brief shout out to the 1980s. Now, these are different times. Some of us in this room weren't born yet. Sherry Krams was doing groundbreaking work as a DJ in the tri-state area. It was a different world, but it was a good world for vaccines in certain ways. Namely, we can think of hepatitis B as an example. So hepatitis B is a virus that doesn't propagate well in culture. So there has to be some other way of vaccinating against it. So Maurice Hillman found that if you took blood from infected individuals, purified it to remove the virus and remove other infectious components, you could get these subunits, hepatitis B surface antigen. And if you immunize someone with an inactive surface antigen, maybe Maurice himself, you could protect people against hepatitis B. So this was the first subunit vaccine in the early 1980s. There was only one problem with using this approach. It was the 80s, and this was the beginning of really the HIV pandemic that we know. So people were uncomfortable with the idea of having a vaccine that was derived from blood products because they were worried that maybe you could pick up HIV that way. Was it really a safe thing to do? So once again, Maurice went back to the drawing board, and he took advantage of genetic engineering to express hepatitis B surface antigen in yeast. So this is what we know as a recombinant vaccine in 1987. So the second point from the 80s that we can take away in terms of vaccine design is the idea of having protein conjugates. And so the idea here is that a lot of bacteria are coated in polysaccharide capsules, sugary capsules. And the problem with that is that these capsules really aren't that immunogenic. I mean, they can bind to B cells, stimulate humoral responses, but we know T cells see peptides in the context of MHC, so you're not getting a full, robust immune response to polysaccharides. So how do we deal with that? So some investigators found you could conjugate or link up proteins, which are immunogenic, to less immunogenic polysaccharides, and the B cells could bind, internalize those, digest the proteins, break them down, and present them to T cells. And this way you get both an antibody response as well as a more robust T cell response. So this was the basis for a vaccine against Haemophilus influenza, which can cause meningitis in certain situations. So here's kind of where the timeline stands, right? We started literally almost thousands of years ago, worked our way into the first vaccine, taken that into the laboratory, learned how to manipulate subunits of a pathogen as opposed to taking whole microbe, and also taken advantage of our ability to grow cells in culture, our ability to link non-immunogenic molecules up to peptides. So we're kind of finishing up the 20th century. And I present you this really simple story of glorious success after glorious success. So everything else from here should be a slam dunk, right? I mean, I hope so. I'm about to be born in 1991. I don't want to have to deal with any of these things. Turns out that's not the case, and you guys already knew that. Malaria, HIV, TB, influenza, hepatitis C. There are any number of diseases for which we either don't have good vaccines or we don't have vaccines. So something went wrong here, and we have to think about exactly what. And one idea, and this is really a good review that's worth looking at, is that the diseases we were vaccinating people against in the past were actually the low-hanging fruit. They were pretty easy to be protected against because the antigens didn't vary that much, and the key was eliciting an antibody response. So really where we've struggled these days is with hypervariable pathogens that require a T-cell response. So we had a difficult time transitioning from this space of pathogens down to diseases like HIV, malaria, and tuberculosis. But the issues that we face aren't just scientific, they're also societal, because we've seen the anti-vaccine movement sort of reemerge in a different form. It's no longer that people are becoming cows, it's that people are becoming autistic. And so as a result, there's a lot of misinformation and fear on the subject that we'll talk about. So if early vaccine design was a Star Wars movie, I think it would be Episode 4, A New Hope. Just glorious success defending the good guys from the evil galactic empire. And maybe for a time recently, we found ourselves in The Empire Strikes Back, screaming with our lightsaber, clattering off into the dark abyss, trying to wrap our minds around how Darth Vader might be our father. So we fell on hard times for a little bit. But I want to bring things back into balance, and I want to do so starting with the facts. So towards the end of the 1980s, 88, 90, there were a couple papers suggesting that the Marizol, this mercury-based preservative, was associated with increased risk to autism. Those studies have all been retracted. People have done larger scale studies showing that's completely inaccurate, and there's no support for that. The Marizol isn't even used in most vaccines anymore. It's used maybe at trace levels. Having said that, autism is still becoming more frequent, so that's clearly not what's going on. So you'd think maybe on some level that a scientist making these types of empirical arguments would work. But common sense and life experience suggests otherwise. So for example, just with a simple example, you have a 1 in 20 million chance of dying in a terrorist attack. Every person in this room, on average. On the flip side, you have a 1 in 4,000 chance of dying by choking on food. People are more afraid of terrorists than they are of peanut butter, right? There was a war on terror. We never occupied Skippy's headquarters. So sometimes numeric arguments don't always add up or don't always resonate with people. Another argument that's commonly made, though, is, I didn't get vaccinated and I was safe. So clearly vaccines don't matter that much. But it's really the success of vaccines that allows some people to be protected, even still, through herd immunity. It's a pretty simple concept, and I'll try to explain it in this cartoon. So imagine you live in a village, right, and no one's been vaccinated. Some people are sick, some people aren't, but there's no immunization right now. So what can happen, of course, is that people who aren't sick can pick up the disease, and you can have these outbreaks and endemic increases in the frequency of whatever the infection is. So the whole point of vaccines, as you guys know, is that if we immunize people and prevent them from getting infected, they don't have to suffer whatever the consequences of the infection are. But what that means for people that decide not to get vaccinated for whatever reason is that they also indirectly benefit, because there are fewer people who can get sick, so there are fewer people who can get them sick. And so this herd immunity, this sort of communal immunity, is what allows a certain fraction of people to not get vaccinated. It depends on a lot of variables, but this is the basic idea. Now, if everybody thought that they could get away without being vaccinated, we now find ourselves back in the outbreak scenario. So it's sort of self-defeating logic in a way. But I want to get back to science for the last couple components of this presentation and leave you guys with some ideas that I think are worth getting excited about in the field of vaccine design. Namely, how we induce stronger T-cell responses. So there's an idea called prime boost, where you can initially take DNA encoding whatever antigens you think are protective, and allow those plasmids to be taken up by antigen-presenting cells, have those genes expressed, presented, and displayed to the immune system. And people have found that when you combine that with viral vectors that express the protein form of these plasmids, or of these genes, that you get even more robust T-cell responses. So this would be in situations where we really need CD4 and CD8 cell immunity. So this is something that's really excited people in the field of HIV. And I want to show you a paper where some researchers tried to take rhesus macaques, challenge them with simian immunodeficiency virus, or SIV, and they wanted to essentially see how many challenges you needed until you can get all of these monkeys infected. So to the point where 0% of them are uninfected. So here we have our mock immunized monkeys, and you see in about 7 challenges, they're all SIV positive. But if you prime boost with a combination of SIV genes, and then you come back with the protein, and just for good measure, you throw in IL-12, the Th1 polarizing cytokine for cell-mediated immunity, that you actually improve the protection outcomes. So this is potentially an exciting way to get T-cell responses where you haven't seen them before. So on the other end of the spectrum, in the antibody world, people are thinking about really reversing the way that we approach vaccine design. So you probably saw this historically. We were kind of throwing everything at the wall and seeing what stuck. But maybe we can reverse engineer vaccines by looking at natural infection and seeing what people are already doing. So the idea is that if you look at infected people, and for example, this is true in the HIV world, there's a subset that produce these really powerful, potent, broadly neutralizing antibodies. So you can isolate these antibodies from a cohort of patients. You can then characterize them on different levels to see how they interact with the antigen. So that can be thermodynamics. You can crystallize the antibodies. You can mutate the surface of their antigen to see what residues are critical for neutralization. And the idea is to collect a pool of information that you can use to work your way back to an immunogen, or maybe a set of immunogens that can mediate the same type of protective immunity that you saw in patients, but do so before they became infected. So this is reverse vaccine design. And I like it because it's a pretty simple concept to wrap your mind around. It raises some interesting questions, like how the hell do you actually do this? So I worked in a lab back at Scripps as an undergrad that was really good at the first couple steps. They were really good at getting these antibodies and characterizing them. So it was Dennis Bird and Dennis Scripps researchers too. But what was difficult was thinking about how you were ever going to design immunogens to pull out those types of antibodies, pull out those types of B-cell receptors. So I want to share with you some ideas on how you can do that. From another group at Scripps, this is Bill Sheaf's lab. So essentially what they did was they studied this class of BRCA1-like antibodies. So these are antibodies that can neutralize, in some cases, up to 90% of viral strains that are in circulation. And many of these antibodies are variants on a common theme. They're somatic variants of one another, and they have a common germline origin. So they're different shades of the same color. And so Bill Sheaf's lab tried to infer the actual sequence and nature of that germline antibody, and then use computational tools to predict what mutations on the surface of HIV would bind best to B cells with receptors that are BRCA1-like. They then tested these mutations by expressing them in yeast, and went back and forth refining that process. It wasn't an entirely rational process. You had to do a little bit of random mutagenesis just to tweak the binding. But ultimately, they found a resurfaced or a modified GP120, which is HIV's surface glycoprotein that's necessary for infection. And they were able to express that on the surface of nanoparticles. So the author of the paper I'm about to show you, Joseph Jardine, is a postdoc in Bill Sheaf's lab. I worked with a postdoc who was collaborating with him, so I kind of knew what he was doing. I didn't realize that it was going to work to the extent that it did. So he found that you can make these 60mers, these nanoparticles coated in 60 units of this modified GP120 core. And then when you did that, you were able to selectively and effectively activate B cells that either express the full-blown mature BRCA1 or the germline version of that receptor. And so here they're measuring calcium flux. in B cells that have been transfected with these different antibodies. So I think this is going to be potentially a really powerful tool going forward, because if we can pull out antibodies that we think could be protective based on their breadth and potency, maybe that means you can take the immune system and kind of steer it in the direction that you want it to go. So I just gave you a very topical overview, but there's a talk coming up that you should definitely listen to. None other than Gilroy's native son. Justin Gerald is going to be talking more about HIV neutralizing antibodies. So I think there'll be some good information there. And now I just want to wrap up briefly by talking about how we can use systems immunology and systems biology to get a better sense of how vaccines work and why they work. So those of you who are on that track in the course are really well poised to make these types of discoveries. So the study I'm going to present is one in which they used yellow fever vaccine as a classic example of an effective vaccine. It induces protective responses for up to 3 decades. So if we can figure out how yellow fever works, maybe we can get clues as to other vaccines and other vaccine efforts. So 20 people, 2 groups of 10, were vaccinated. We collected peripheral blood cells from these people and ultimately did microarrays, Luminex, and flow cytometry to look at transcription, cytokines, chemokines, and different cell subsets. So the researchers in this paper, so this is Carrick et al., came out in Nature Immunology, found that you could use principal components analysis, take this really complex microarray data, and distill it to a form where you can see that people with efficient CD8 T cell responses to the vaccine cluster separately from those who don't. And so they were able to identify genes that were important for this differential CD8 T cell response. What they then showed to validate this result was that when you took the genes that were important for the segregation and you took responses from one group of 10 people who were vaccinated and used those genes to predict the responses in the other 10 people who were vaccinated, you got a fairly low error rate if you use between 20 to 30 genes. You're probably wondering what these genes are. Some of the ones that came up were genes for complement. There were others that weren't really as well understood, including glucose transport. It's not immediately clear how that would have been relevant to the CD8 T cell response. But this is maybe one of the beautiful things about the approach. You can see things you weren't looking for, go back to do bench studies, and ultimately find what people haven't found in the past. So these are definitely ideas that are worth looking at in the future. If you guys hear talks on either systems immunology or reverse vaccinology or prime boost studies, worth checking out. So I want to wrap up by leaving us with something that's pretty relevant, how we as immunologists can contribute to vaccine design. Because up until recently, vaccine design was empirical, it wasn't entirely rational. So I think we can make an impact in a couple of different areas, namely identifying correlates of protection, aspects of the immune response that are most important and that are indicative of a successful response to a pathogen. And so the systems tools that I just talked about are going to be useful for that. And maybe they'll suggest certain pathways that we need to target and that we can develop adjuvants to, right? So using tapioca and breadcrumbs is probably no longer a tenable strategy. So we understand that the innate immune system is critical, and we know the receptors involved, TLRs, NLRs. And so we can start to target those, and people are doing that already. And ultimately, any vaccine has to get to where it needs to be. And there was a great talk by Daryl Irvine last Friday, among other things, dealing with how we get adjuvants and immunogens to draining lymph nodes. So that's going to be critical going forward. But in the grand scheme of things, I'm feeling pretty good about vaccines' future, because we're the future. And I want to thank you guys for listening to my presentation, for all the feedback. Thank Lakshmi, thank the TAs, Thomas and Trini, and thank Sherry. And it wouldn't really be a presentation of Wosen's if I didn't have some alliteration somewhere. So, enjoy.
So you're at about 34 minutes and 30 seconds.
Mm-hmm.
So we'll open it up to content-based questions.
With the 60 mer
Yeah.
GP protein name engineered, have they been able to actually demonstrate that those particles drive the production of similar rearrangements?
Not yet. Yeah, not yet to my knowledge. I mean, it's pretty still in development, but that would obviously be the next thing. One of the next things you want to do. Yeah.
So, I mean, I don't know much about adjuvants and what you use it for, but do you have any example of like adjuvants that stimulate TLRs or?
Yeah, so I mean, people are using
like CPG.
Yeah, CPG, poly-IC. I don't know. Some of these have been given the go-ahead for clinical use, and others are just used in the laboratory right now. So, yeah, the process of discovery and then actually getting it to a point where it's being used is slow. One of the weird things, I don't know much about adjuvants either, but it's not clear that anyone does, because one of the oldest adjuvants, alum, was developed in 1926. People still don't really know how it works. There have been different theories proposed, most of which have either been struck down or have a lot of caveats associated with them. Alum itself doesn't actually get taken up by the cells, but it seems to promote antigen uptake in a way that doesn't involve phagocytosis. It's very confusing, and actually alum itself is one of the reasons why we probably haven't gotten strong T-cell responses in the past. So people have shown that alum, through apparently the ERK signaling pathway, lowers levels of IL-12 expression in dendritic cells, so it's skewing us away from Th1. So that might have been historically one of the reasons why we saw predominantly antibody humoral type responses. Justin?
Do you know if there's been any movement or more improvement in influenza vaccine generation? Because we've been using the egg model for so long, and it seems to be laborious and still limiting. Do you know if there are improvements that are currently being made to make it more feasible work?
That's a good question. So I don't know. I'm sure they're out there if I look sort of more thoroughly through the literature. Mm-hmm. I know that historically there's been some effort to think about like, influenza and HIV vaccine development sort of in tandem, just because of some of the similarities in how those viruses work and the idea that they're both highly variable. If you can come up with a vaccine to one, you'll probably know a lot about vaccinating against the other. But yeah, so I don't have anything necessarily for you on that point, but I'm sure it's out there. Yeah.
Why the spines of bunnies for rabies?
Why? So I think it travels through the cerebrospinal fluid.
How did they know? Like to use? Was it just because they tracked how it traveled, or how do they know to use the spine of bunnies? Like I guess why not dogs or something? I don't know.
Yeah.
I guess dogs would be holding them, right?
Yeah. Oh, I mean, they used rabbits because that was their model in the laboratory. I mean, maybe they had dogs that they were testing as well. I think they did, but yeah, ultimately as to why the spinal cord, I don't.
Rabies travels through the neurons, so
okay.
it's a tropism, right? It's like the same as HIV with the macrophages and T cells.
Okay.
Yeah.
That's why if you get bit, you need to get the vaccine before it reaches the brain.
Yeah. There's a Radiolab episode on this, like the one that the girl.
Yeah.
Like they induced coma to a girl and then she like survives rabies.
Crazy.
Like this, it's really good episode. I like it.
Okay, I won't spoil it for you, so I'll just let you get it.
Okay, sorry, I totally spoiled it.
No. It's good.
Sorry.
No.
So you showed the really nice figure where we have vaccines for things that produce good antibody responses and the ones that we don't have vaccines for. Similarly, you need good T-cell responses, and you offer a solution, or people offer a solution where you can prime DCs with the DNA encoding the antigen. Are there any studies that show either trying to target limited diversity of the viruses or pathogen? And do you know how successful they are at presenting a common antigen?
Yeah. So I don't know. Trying to think. Can you repeat the second part of the question, actually?
Yeah. So when they prime the DCs with the plasmid encoding the antigen, how successful are they at making T-cell responses to a virus that changes so frequently, or is it just to that one type?
That's a good question. I guess one way to maybe have looked at that, if I had looked into, in that SIV figure, whether they challenged with just one strain, whether they actually varied the strains, maybe that would tell you something about how broadly the protective response was induced. So, there may be some clues in there somewhere. I don't know for sure.
One more kind of a very, I don't know if this is something that's well known or not, but we hear a lot about drug-resistant pathogens. Is it similar where you can be vaccinated with something and there are like vaccine-resistant strains, or are most of them pretty pan? And how do you mediate those effects, and how can you get more broadly applicable vaccines?
Yeah. So, I mostly understand vaccine resistance in the sense that if you have highly variable viruses, you can't pick just one strain, and oftentimes conserved regions. So, for example, there's that part of HIV that binds CD4. So every strain of HIV has to have that. But it's buried underneath this thick forest of glycosylation. So, maybe some strategies can revolve around, sort of artificially expressing forms of the virus that are more accessible to the immune system. In some cases, we know that some of those glycosylation marks are actually necessary, though, for the neutralization of antibodies. So it's a complicated story in terms of how you actually get those responses to work.
So along those lines, could you talk about how we predict and prepare for, say, the next flu epidemic or whatever? Like, how do we plan flu vaccines?
Yeah. So I think there are a couple of different things that go into it. There's a paper that came out earlier this year that I missed. I know Steve Schaefer gave a talk on it, Postdoc Club. But one of the things that goes into account are what the escaped variants were from the previous year. So what was the strain of the flu everybody got last time around? That can tell you about what's going to come back. But I think these days there are some in silico things you can do as well to predict what are going to come up as mutations. And that probably takes into account the fitness cost of particular mutations, as well as the increase in virulence that you would get. That's the level to which I know. Yeah.
I did have another question. Do you know the transition between the use of side chain to antibody came about, or how did that come about?
So I think it was first it was a transition from side chain to receptor. I don't know when the term antibody itself came into being. Maybe something else.
I think it's from antitoxin.
Yeah.
I forget exactly what, but it's originally from antitoxin, I think from diphtheria. They're looking at rabbits and antitoxin in serum.
Okay. Are there any other additional questions before we move on to the feedback portion? Okay. So any thoughts on design?
So beautiful. Like, everything was so just perfect and easy to follow and well designed. It was just honestly like watching a movie. I feel like I was that entertained. It's really good.
Yeah, I tried to pay a little bit more attention to the visual aspect this time around. So hopefully that can be awesome.
I really like it too. I think you definitely followed the advice that we gave you last time, because you're a great speaker. You don't need a lot of text in your slides because we already follow you. So it's good because whatever was in the slide was not it was good because it guided the audience to what you were talking about, but it didn't distract us from listening to you because we weren't reading a lot there. Yeah. But and then you also, I noticed at least that you also made most of the diagrams and stuff, which is good because in a way there wasn't anything on the slide that you were not going to mention, right? And that's always good for us to keep focus. So.
Yeah, from a design standpoint, going along their two comments, it reminded me a lot of these educational videos that you might find on YouTube on on specialized topics, and I really appreciate that. So the whole time that you were delivering this, I kept thinking about, this is something that can be useful for high schoolers to watch, just because there's a high level of detail, but then you present it in a way that is accessible to anybody, and I think that's really good science communication.
That like comment, I think, transitions to both the delivery and content and visuals, which is that you had something that was really simple and elegantly put forward that and always kind of just funneled to like cutting edge research really nicely. And so it was just on all three criteria with both the content you picked, the way you delivered it, and then your just actual visuals associated with it. It worked really nicely. And so I didn't have any like harsh transition. I was like easy, fun, easy, fun, science. It was just nice. Yeah.
Any other thoughts on design before we move into delivery? Okay. Any thoughts on delivery? Additional thoughts?
You're such a great speaker. Mm-hmm. Like, honestly, you should do this for a living. Like,
I think you're better than everyone that presented in Advanced 2, for sure. Or Advanced 1. I forget what it was. Advanced 1. You were better than every single professor that we had, honestly. Sorry.
You know?
Oh, yeah, you should give this for the vaccine shot in Immuno 1.
Yeah.
I'll think about it.
I think you have great pace, and that's stuff that since you're so concerned with alliteration, I think that makes you very conscious about your vocal variety as well as your pace, and I thought that they were on point.
Yeah, I knew it would come in handy one day.
Yeah.
It was nice seeing you practice yesterday and then see the final product, because like this is like the pure, smoothed out, facial, really, I don't know, sparkly, shiny.
Yeah. So I practiced yesterday in like room 106. So you can rent out the room, or you can reserve the room if you want to practice your talks.
Oh, I saw you coming here. I was outside.
Oh, okay. Were you talking to a friend?
I was talking to a friend, yeah.
Yeah, so if you guys want to practice either this room or 106 are usually available. I guess it was like watching the bootleg version of an actual movie.
Yeah, it was like watching the bootleg version. Yeah. It was there, but some of the translations were just a little jerky. But like you put a lot of work into making these good.
That's usually the case for good talks. You can tell that they spent a lot of time on it. If you go to conferences, you'll see when they didn't spend any time at all. They just typed it up right before they went up there to present. In any case, any additional thoughts on delivery before we move on to content?
It's really nice that your presentation just sounds like an anecdote, like a story, and you don't really get that in science. Mm-hmm. And I don't, like many talks that I've gone to, I feel like I'm struggling to understand it all the way through. Or I'm always like a couple of steps behind, and then I just give up because I'm like, I'm never going to catch up. But this one is just like, I was excited about every new slide that I was going to see, and it just kept on building and building. It was really good. It's probably one of the best presentations I've seen.
And then you put his face in there. So
well, yeah.
Yeah,
I put a lot of pressure on that. But no, it was amazing.
Yeah, I tried to think a little bit about. There was that graph we saw from that speaker who spoke about speaking with like the different drop-offs and attention. Yeah. So I thought a little bit about where I could insert things that would sort of bring that back up periodically.
I think the fact that you were very audience-centric shows, which is why we paid attention to the entire presentation.
The timeline helped. Yeah. That was really nice.
Okay. So we're moving on to content, I guess more specifically. Any thoughts on content? Additional thoughts?
Would have been nice to see even more primary data.
Yeah, I think so.
Yeah.
But hey, you had the right time taking up. So.
Yeah. It's always a hard balance when you're doing these type of historical talks that cover 1,000 years. It's like, you kind of want to give people an overview, but then you want to give them a little bit of depth, but then having to come back to the big picture takes up some time, and you only have 40 minutes. Yeah, I agree. Some parts, I guess, you did have a few primary data slides, especially the part where you discuss your scripts work. And I like how you insert yourself, so you incorporate yourself into this talk of history, which I think makes it a little bit more poignant and certainly piques my interest a lot more.
Yeah, I think the way it worked was like you start at the beginning historically. You spend a lot of time researching these ancient stories, and then like 1 week or a couple of days before, you realize you actually have to like skip through several centuries worth of work and get to the actual like current events. So
Yeah.
Yeah, it was a little heavy on the early stuff, but
Sounds familiar.
yeah.
I think what would have helped with the content, I really love the timeline, which is the comment I was going to make with your talk. Afterwards, I completely forgot. I think that's useful for everybody here if you're going to present it in a chronological fashion. I think would have helped in the second part is after you got to 2000 if you had some other type of road mapping.
Yeah.
Because I wasn't sure what direction or where we're headed in this conversation when you were presenting. Any other thoughts? Good job. Thanks.